Hello, in this video I'll be covering what beta-amyloids are, their types, functions, and distinction between monomeric and oligomeric beta-amyloids. Beta-amyloids are relevant to Alzheimer's disease as they are one of the main two hallmarks of Alzheimer's disease. Also note that this is a part 3 of a series of videos that investigates the true nature of Alzheimer's disease. You can find the other videos and supplementary information in the description below. So one theory of Alzheimer's disease pathology is that beta-amyloid plaques keep accumulating in the brain to cause Alzheimer's disease. But what are beta-amyloid plaques? Well, first of all, a beta-amyloid is a small peptide or protein that has a long, slender, fiber-like structure, fibril for short. And beta-amyloids are peptides that make up the majority of a beta-amyloid plaque. Other than a core of amyloid, these plaques are also composed of fragmented and decaying nerve terminals. What usually happens is that if the plaque gets too big, it starts pushing on the nerves around it, causing the miscommunication and eventually damage to those nerves. Beta-amyloids is a protein that the neurons secrete. Even healthy people's brains secrete beta-amyloids, but the problem is that these beta-amyloid proteins are not cleared away properly in Alzheimer's disease. That's why beta-amyloids keep building up in the brain of Alzheimer's disease patients to form up the plaques known to cause Alzheimer's disease. Beta-amyloid plaques do build up outside of the cells and block the neuronal communication by that. But there is a reason why these proteins are secreted. Beta-amyloids, by another school of thought, is thought to be a protective, reparative, and restorative, and a defense mechanism against Alzheimer's disease. So in Alzheimer's disease, the problem is not necessarily that these proteins exist, but that these proteins aren't being cleared away properly. So if the person asks, where do beta-amyloids come from? One may say that beta-amyloids are produced when something called the amyloid precursor protein, or APP, is sliced by an enzyme called the beta secretase and gamma secretase. So theoretically, an increased activity of beta and gamma secretase increases the production of beta-amyloids from the available APP, thereby increasing the risk of developing Alzheimer's disease. But this depends on the type of beta-amyloids produced. That's because there are many different types of beta-amyloids, with beta-amyloid 40 being relatively benign and beta-amyloid 42 being quite destructive. So the genes that are associated with an increased risk for Alzheimer's disease encodes the APP protein, and the APP is ubiquitously found in the neural and non-neural cells. So beta-amyloids don't refer to only one homogeneous molecule. Rather, there are many different types of beta-amyloids, and the number used to differentiate the types of beta-amyloid simultaneously refers to the length of the beta-amyloid peptide, or how many amino acids the amyloid beta peptide is made of. So for example, a beta-amyloid 40 is composed of 40 amino acids. Amyloid beta 40 is the most common species of amyloid and is relatively benign. Amyloid beta 40 does not lead to plaque formation and in fact protects against plaque formations caused by the deposition of beta-amyloid 42 in the brain. And on the other hand, beta-amyloid 42 enhances the formation of oligomers, which is detrimental to their neuronal health. What happens is that the beta-amyloid 42 initially forms microscopic deposits in the brain. These deposits are diffused plaques, diffused meaning not fibrillary. Then the diffused plaques trap strands or fibrils of beta-amyloid, thereby becoming a fibrillary plaque, which then causes localized inflammation around those fibrillary amyloid beta plaques. The inflammation is characterized by an increased level of white blood cells such as microglia and astrocytes, and those white blood cells release free radicals as a part of the inflammatory response. This causes collateral damage by reducing the number of synaptic spines or projections, harming the surrounding neurons and eventually killing those neurons. Amyloid beta molecules can group together to form flexible soluble oligomers. Oligomers are a complex of molecules that are made up of a few repeating units. The theory that accumulating beta-amyloids causes Alzheimer's disease also states that those misfolded oligomers of amyloid can act as seeds, causing other molecules of beta-amyloids to form misfolded oligomers. Like this, amyloid beta oligomers, aka amyloid plaques, may cause a chain reaction for inducing brain dystrophy like a prion infection. Oligomers of amyloid beta can be more damaging than plaques of amyloid beta because fibrillary plaques have much less surface area to neuronal membranes. In contrast, oligomers are higher in number and can diffuse or pass through the synaptic clefts of a neuron, causing damage from within. So oligomers are more likely to cause neuronal synaptic dysfunction. Indeed, the quantity of soluble oligomers correlate better with cognitive decline than the plaque count. Single molecules of amyloid beta or monomeric amyloid beta are digested or broken down by an enzyme called insulin-degrading enzyme, or IDE. But oligomeric amyloid beta is resistant to be broken down by IDE, or insulin-degrading enzyme. Amyloid plaques tend to be heterogeneous mixtures of 37 to 43 amyloid beta peptides. One function of an amyloid plaque is to sequester the toxic amyloid beta oligomers that destroy the neurons that it interacts with. Although amyloid plaques causes the surrounding neurons to become dystrophic, it overall reduces the damage that separate oligomers of amyloid beta can cause. Initially, it was proposed that amyloid beta plaques in general were the main driving force of Alzheimer's disease. So accordingly, what increases the amount of amyloid beta theoretically would be the main cause of Alzheimer's disease. For example, in one study, scientists found that the mutations in presenilin 1, presenilin 2, and the amyloid beta protein precursor proteins genes increases the extracellular concentration of amyloid beta 42 to 43. Note that the mutations in these genes are all linked with the incidence of familial Alzheimer's disease. So this indicates that gene mutations that increase the production of beta-amyloid may cause Alzheimer's disease. Theoretically, what stops the clearance of amyloid beta from the brain could also cause Alzheimer's disease. Note that the type of beta-amyloids make the poison. For example, beta-amyloid 40 is relatively benign, whereas beta-amyloid type 42 or 43 is rather toxic to the neurons in the brain. The amyloid beta oligomers are toxic to nerves, not necessarily single independent units of amyloid beta itself. So some studies even show that the abundant amyloid depositions in the humans and mice brains do not necessarily show memory deficits or neuronal toxicity. In fact, the presence of separate amyloid beta molecules may have a beneficial effect on the surrounding nerves. Before I elaborate, note that an absence of amyloid beta is not observed to cause a loss of brain function, but if present, amyloid betas, especially amyloid beta 40, functions as a highly potent antioxidant that protects against free radical oxidative stress, and the beta-amyloid plaques can be beneficial by means of damage reduction, sequestering the damaging amyloid beta oligomers that cause neuronal synaptotoxicity. In another study, amyloid beta 40 monomers were seen to be beneficial by acting as antioxidants for free radicals generated by different types of metals in an unhealthy way. Usually, the increased concentration of different types of metals in the brain occurs over time. That may be the reason why cognitive decline is observed with advanced aging. The different types of metals that gather in the brain include heavy metals such as lead, mercury, and cadmium, and other metals like aluminum. And that's the end of part 3 of the Alzheimer's disease series. In part 4, I'll teach about the tau tangles, which are the other main hallmark of Alzheimer's disease. Thank you for listening. If you have any questions, please feel free to ask me in the comments below. I'd appreciate if you like and share this video, and subscribe to my channel. Bye!